Hello students, how are you all? I hope you all are well prepared for 2025 Chemistry Cbse final examination, that is in class 12th. Now on 27th you have the exam. I hope you all are well prepared. I have some suggestions for you for biomolecules, that is the last chapter of your you can take in this one as biochemistry portion. So you have 7 marks in the chapter. So I will tell you some important topic that can directly come in the exam. So with 4 or 5 topics you can see and you can get the marks from there. Last year from biomolecules chapter there is a study based question, case study based question you can say. So that was given from DNA and RNA. Now this year if there is any kind of case study based questions given from the DNA biomolecules chapter, that may not be from DNA RNA, that can be from the glucose portion, that means from the carbohydrate portions or from the vitamin portions. There is 100% probability from the glucose portion if it is any kind of case study based question is given. Okay, now we cannot predict like that, right? So what you have to do, we have to go with those topics by which we can easily get the marks. Now here we can easily get 7 out of 7 and we have to get 70 out of 70, we have to get 7 out of 7 from here. Is it clear? So now one thing is that the carbohydrate is the very important topic here. Now under carbohydrate, glucose is one type of what? Monosaccharide. Yes. So that glucose, that means the D-glucose, takes part in chemical reaction with many chemical reagents. So those reactions are very important. Each and every year they give 2 or 3 reactions, either in 1 mark question or in the any kind of MCQ they can give in assertion and reason also. So I'll tell you 5-6 chemical reactions with D-glucose that you have to follow, and the balanced chemical equation you have to write. Okay, first thing is that glucose with zinc amalgam and concentrated cell. Okay, the first thing is that glucose with zinc amalgam and concentrated HCl. Number 2, glucose with concentrated nitric acid, and it will oxidize to form saccharic acid, right? We have learned. And in the previous one, from zinc amalgam and concentrated HCl is a reducing agent, and that will reduce the aldehyde group of glucose into CH2H. Okay, so that portion is very important. Glucose with bromine water, glucose with HI means hydrogen iodide, glucose with hydrogen iodide in presence of red phosphorus. Okay, 2 products will generate. The 2-iodohexane will be the major product that you have to write. Okay, next is that you have to write the reaction with HCl, that means the formation of cyanohydrin. The cyanide that nucleophile cyanide will attack the carbonyl carbon of the aldehyde group present in the glucose and will form the cyanohydrin, that one. And the reaction with excess amount of phenylhydrazine, glucose with excess phenylhydrazine, that reaction is not given. They have never given this kind of reaction, so you cannot trust this will be not given, that will not be given. So you have to prepare all these reactions. Okay, and again one more reaction, that is glucose with means acetylation of glucose, you can say, with acidic anhydride, and that also confirms that glucose has 5-OH group, because all the hydrogens of CHOH will be replaced by that acetyl group and will get a product. So these reactions are very important. Now next is that from protein, denaturation of protein, renaturation of protein. Okay, primary, secondary and tertiary structure of protein. Okay, alpha helix structure, beta pleated sheet structure, very important, that can directly come in the exam. Okay, again as I mentioned, that can be also can come as a case study based question. Okay, so you have to remain prepared for that. Okay, now from the DNA and RNA, more or less every year they give 1 or 2 questions. Now from the structure oriented, the structure of DNA, structure of RNA, double stranded helix structure, single stranded, the nitrogen bases, adenine, guanine, cytosine, thymine. In case of RNA, adenine, guanine, cytosine, uracil, nucleotide, nucleoside. Okay, then another one portion, the building block of protein. We know what is the building block of protein. The amino acids are the building block of protein. So how the amino acids are structurally built up? What is the structure of amino acid? In each and every amino acid, we know that there is a NH2 group, that means amine group, and there is a COOH group, and there is a central carbon having 1 hydrogen, and the groups will be replaced and will get different amino acids. So for that, glycine, leucine, lysine, phenylalanine, different type of amino acid structures you have to learn, and from there the Zwitterian structure of amino acid, very important. Previous year Cbse never asked the formation of peptide bond. They have given what is a peptide bond, like that question was given. They can ask Ella, Gly, Aspa peptide bond structure. So you have to remain prepared for that, how to draw a peptide bond structure with different kinds of amino acid. Okay, optically active, optically inactive amino acid. Only one amino acid is optically inactive, that is glycine, because it has sigma plane. Other are optically active. Okay, Zwitterian structure of glycine, that can also come. Basic amino acid, acidic amino acid. Which one is basic, which one is acidic? The amino acids having more number of COOH group, that is acidic amino acid, like aspartic acid, and the amino acid which has more number of NH2 group, they are like basic amino acid, you can say. Amino acids are normally amphoteric in nature. Why? That also very important. Okay, monosaccharides, polysaccharides. Every year there will be one question from the monosaccharides, polysaccharides. So examples are also important. Okay, glycosidic bond. The glycosidic bond is a very important topic for Cbse. Every year I have seen either in the assertion region or in the MCQ or in 1 mark question it is given. So remain prepared for that also. For glycosidic bond, anomeric carbon. Which one is the anomeric carbon? What are anomers? And mutarotation of glucose. What is mutarotation? What is anomeric carbon? What is anomers? What are anomers? Give examples. Okay, how are structure of glucose? Again, the structure of that pentose sugar, that is also very important. Pentose sugar, then along with phosphate group, you know, sugar plus base is and again sugar plus base plus phosphate group is so there you know about DNA and RNA, so those portions you have to learn. So pentose sugar structure, then from there adenosine, nucleoside structures, those things you have to see again. Okay, vitamins, C, vitamin B, what are the effect of deficiency of those vitamins? Okay, water soluble vitamin and the oil soluble vitamin, or you can say fat soluble vitamin. Their deficiency is important for this year, because last year I have seen there was a question with what are the water soluble vitamin, what are the example of fat soluble vitamin. So this time they can ask the deficiencies, scurvy and all, ricket and all. So these things you have to see. So all the best for your 27th exam. I'll make another video for one shot with all the organic chemistry chapters important topics. Okay, and I'll make you understand some important name reactions also, and that can easily come in the exam. Okay, so all the best for this chapter. Remain prepared. You have one day again before 27th. All the best.